Saturday, September 9th, 2023, AGNDD Intellireport topic. Serotonin. Please pardon any visual and informational inaccuracies in this entirely AI-generated Wikipedia-sourced video article.
The indoleamine molecule derived from tryptophan undergoes biochemical reactions to form serotonin. Serotonin adopts different conformations and forms hydrogen bonds, playing a crucial role as a neurotransmitter and in pharmaceutical formulations. The indoleamine molecule is derived from tryptophan through a series of biochemical reactions. The hydroxylation of the 5 position on the ring forms an intermediate called 5-hydroxytryptophan, which is then decarboxylated to produce serotonin. The molecule can adopt 6 different conformations, with the preferable conformations being defined by the ethylamine chain. Serotonin, a neurotransmitter, crystallizes in a chiral space group called P2-12121. It forms hydrogen bonding interactions between serotonin molecules through NH.O and OH.N bonds. Serotonin can also form various salts, including serotonin adipate, which is used in pharmaceutical formulations. Serotonin plays a role in various physiological processes such as sleep, learning, behavior, and feeding. It also regulates processes in less complex animals and is associated with stress signals in plants. However, the belief that low serotonin levels directly cause depression is not backed by scientific evidence despite its promotion in pharmaceutical advertising. Serotonin acts through its receptors, and its effects vary depending on the cells and tissues that express these receptors. The metabolism of serotonin involves oxidation by monoamine oxidase, followed by hydride transfer to the flavin cofactor. This is the rate-limiting step. The oxidized form is then further metabolized by aldehyde dehydrogenase to form 5-hydroxyindoleacetic acid, 5-HIAA, which is a derivative of indole acetic acid. Finally, 5-HIAA is excreted by the kidneys. The 5-HT receptors are found on the cell membrane of nerve cells and other cells in animals. They are responsible for mediating the effects of serotonin, as well as various pharmaceutical and psychedelic drugs. These receptors can be classified as either ligand-gated ion channels or G-protein-coupled receptors. The activation of these receptors leads to the initiation of intracellular signaling pathways. Serotonergic action is primarily terminated through the uptake of serotonin, 5-HT, from the synapse by the monoamine transporter SERT. However, a study suggests that a newly discovered transporter called PMAT may also play a role in 5-HT clearance. PMAT has a lower affinity for serotonin compared to SERT, but has a higher transport capacity. Some SSRIs inhibit PMAT, but at concentrations much higher than therapeutic levels. PMAT may also transport dopamine and norepinephrine, but at even higher concentrations. No known pharmaceuticals significantly inhibit PMAT at normal therapeutic doses. Serotonin can signal through a process called serotonylation, where it modifies proteins. This process is involved in serotonin's effects on platelet-forming cells and the release of vesicle contents. Serotonylation also plays a role in the pancreatic release of insulin. Additionally, serotonin affects vascular smooth muscle tone by modifying proteins involved in muscle cell contraction. This process is related to the original naming of serotonin based on its effects on smooth muscle. The RAFE nuclei in the brain are the main source of serotonin release. There are 9 RAFE nuclei labeled B1B9, which contain the majority of serotonin-containing neurons. These nuclei are located along the midline of the brainstem, centered on the reticular formation. The neurons in the RAFE nuclei send axons throughout the central nervous system, with lower nuclei terminating in the cerebellum and spinal cord, and higher nuclei spreading throughout the entire brain. The serotonin nuclei can be divided into 2 main groups, the rostral and caudal groups. The rostral group consists of 3 nuclei, while the caudal group consists of 4 nuclei. These nuclei project into various cortical and subcortical structures. The serotonergic pathway is involved in sensoromotor function, motor activity, and intense visual stimuli. Serotonergic activity increases with motor activity, and firing rates of serotonergic neurons increase with intense visual stimuli. Serotonergic projections from the caudal nuclei regulate mood and emotion. Serotonin is a neurotransmitter that has important functions in the brain. It activates 5-HT receptors on nearby neurons and impacts various aspects such as appetite, motivation, dominance, aggression and anxiety, aggression. Serotonin plays a vital role in regulating gastrointestinal function. Enterochromaffin cells release serotonin when food is consumed, which stimulates the gut to contract and propel the food forward.